In this video, we will discuss about the supporting cells of the nervous system. These are called glial cells. What are cells of the nervous system? The nervous system comprises of neurons and glial cells. Neurons conduct signals. Glial cells are non-excitable supporting cells. They provide critical support, maintenance, and protection. Abundance of glial cells. Glial cells, also known as neuroglia, outnumber neurons by a ratio of about 10:1. In the central and peripheral nervous systems. Classification of glial cells. Glial cells are classified based on their location. As there are different types of glial cells in the central nervous system and peripheral nervous system. Glial cells in the central nervous system. There are 4 types of glial cells in the CNS: astrocytes, oligodendrocytes, microglia cells, ependymal cells. Now we will talk about the glial cells in the peripheral nervous system. There are 2 types of glial cells: Schwann cells, satellite cells. What is the origin of glial cells in embryo? There are 3 different sites of origin for specific types of glial cells. Microglia cells originate from mesoderm. Ependymal cells, oligodendrocytes and astrocytes, originate from neuroectoderm. Schwann cells originate from neural crest cells. Now we will discuss the details of glial cells in the central nervous system. Number 1. Astrocytes. Structure. They are star-shaped cells with multiple processes. They are the most abundant glial cells in the central nervous system. Functions. They perform the following functions: support and repair. They provide physical and metabolic support to neurons. Blood-brain barrier. They help maintain the blood-brain barrier by regulating substances entering the central nervous system. Neurotransmitter regulation. They remove excess neurotransmitters from synaptic clefts. Ion balance. They maintain extracellular ion homeostasis, especially potassium. Scar formation. They form glial scars after central nervous system injuries. Clinical relevance. Astrocytomas are tumors derived from astrocytes. Number 2. Oligodendrocytes. Structure. They are small cells with fewer processes than astrocytes. They are found in white and gray matter. Functions. They perform following functions: myelination. They form the myelin sheath around multiple axons in the central nervous system. This increases signal conduction speed through saltatory conduction. Clinical relevance. They are affected in multiple sclerosis, leading to demyelination in the central nervous system. Number 3. Microglia cells. Structure. They are small, rod-shaped cells, with fine processes. They are derived from monocyte, macrophage lineage. Functions. They perform following functions: immune surveillance. They act as the resident immune cells of the central nervous system. Phagocytosis. They engulf pathogens, dead neurons, and cellular debris. Cytokine release. They regulate inflammation in the central nervous system. Clinical relevance. Overactivation of microglia cells can lead to neuroinflammatory disorders, like Alzheimer's disease. Number 4. Ependymal cells. Structure. They are ciliated, cuboidal or columnar epithelial cells, lining the ventricles and central canal. Functions. They produce following functions: cerebrospinal fluid production. They form part of the choroid plexus, that produces cerebrospinal fluid. Cerebrospinal fluid circulation. Their cilia aid in the movement of cerebrospinal fluid within the central nervous system. Barrier formation. They act as a barrier between the cerebrospinal fluid and brain tissue. Clinical relevance. Ependymal cell tumors, such as ependymomas, can disrupt cerebrospinal fluid flow. Now we will discuss the details of glial cells in the peripheral nervous system. Number 1. Schwann cells. Structure. They are elongated cells that wrap around axons in the peripheral nervous system. Functions. They perform following functions: myelination. They form the myelin sheath around a single axon in the peripheral nervous system. Regeneration. They aid in axon repair and regeneration after injury. Support for non-myelinated axons. They surround groups of smaller axons without myelination. Clinical relevance. Schwannomas are benign tumors, arising from Schwann cells. Number 2. Satellite cells. Structure. They are small, flattened cells, surrounding the neuronal cell bodies and ganglia. Functions. They perform following functions: support and protection. They provide structural and metabolic support to neurons, in the peripheral nervous system, nutrient exchange. They regulate the exchange of nutrients and waste products. Ion regulation. They maintain ionic balance in the extracellular environment. Clinical relevance. Dysfunctions in satellite cells may contribute to neuropathic pain,